Welcome to Spring LIB.
Okay, so problem 78 is all about cell size in prokaryotes, right? So the idea here, and we have a little graph in the book, right? If our x-axis is our cell size, and the y-axis is the number of cells. When we take a look at the distribution of cell sizes, sort of at your initial state, there's a small distribution. The idea is, the thought is that a large cell, right, should be able to, during the growth process, become at times more than twice as big. And then when it divides, we'd be able to make two cells that are larger than initially, right? And that the same thing could happen with small cells, right? The small cell might not grow to be more than double its size, and then it would result into even smaller cells. Okay. And so then what you would expect is that in a future time, right, if the larger ones can become even larger and the smaller ones can become even smaller, that your distribution would change to something like this, okay? But that does not happen, so we can just rule that out of reality. And there's some theories for why, right? And so the theory number one that we're given is that there is a timer, okay? And what this means is that as the cell grows, it's only allowed to grow for some number of minutes or hours, right? And no matter how much it's grown in that time, once the time is up, then it will divide. Okay, and then our second theory that we're given here is that there's a volume threshold. So instead, the little cell is growing, and it's going to grow until a very specific size, right? Like a maximum size, and then it will divide. And then I guess I can just use the little image above, right? And then it divides, okay? So these are some of the two main theories. And about 5 years ago, this is 2019, so I guess in 2014, we should say a third model was suggested, right? Model number 3 states that the cells increase in a constant volume. Okay, so constant increase. And what that means is that whatever the size the cell is, if it's, has a diameter of some units, so we have a diameter of n units, then it will always increase. It will increase to a diameter of this like n plus some constant k units instead. Okay, so that would be our new diameter. And so these are the three main theories we're given. And so now we're asked to sort of justify these theories. And we can do so in two parts here. So the first one says to sort of reject theories 1 and 2. And then in the second part, sort of discuss what the impact of 3 would have on our graph, okay? So we can do that here. And if we look at the first two claims, so the first one was having a timer. Okay, having a timer means that as the bacteria would grow, it would sort of be cut off at this particular time, right? It'd only be allowed to grow to a certain point. And so then that leaves the question of, well, what happens when there are limited resources? Okay, because growth is not constant. It depends on nutrients and availability of those nutrients, the ability to take in the nutrients. Space is an issue, right? So you can consider space and competition and population size, right? And so if you're in a limited resource area, you might never even get enough nutrients to divide, right? Because if you recall, you need quite a bit of nutrients for that process, right? So that could cause you if there's not enough nutrients, that once your timer ran out, you were not able to divide, right? So it could actually, having a timer in various situations could actually reduce the cell's ability to even divide, okay? And we can see cell division in limited resource environments, right? So that suggests that that is not the mechanism that we're using. And then the second one, if we recall, is the volume threshold, right? Okay, and this is the idea that there's some maximum that the cells can get to. So we have a little bacterial cell, and it's going to grow and grow and grow. And then once it hits, some sort of diameter maximum, it'll just stop growing there. And then it will divide. Okay, but so to do this would require that at each stage of growth, right? So we get to here, then the cell has to do like a internal check. Am I big enough yet? So I guess it would be, no, I'm not big enough yet. And then it would grow again. It would say, well, am I big enough yet? And the answer would be no. And it would grow again, and it would have to check again. Am I big enough yet? No. And then eventually we would get to this final stage and it would say, okay, great. Now I'm big enough and I can move on. But to do this, to have this sort of mechanism, that would require more work by the cell, right? So that would A, require just having the mechanism, and B, require that some amount of the energy that the cell has go towards checking if it's large enough, right? And that's not really ideal. You want as much energy as possible to go towards growth and reproduction. Okay, so that sort of suggests that this is not really the likely scenario. And so then we get to our sort of part 2 of A, which is to argue about adding a constant volume, right? So this time we're just adding our constant volume. And so this one says that regardless of cell size, if we're a small one or a large one, whatever the diameter is, at the next stage, that diameter will then be this sort of like, so if this had diameter n, this new one would be n plus k. And if our larger cell here had a diameter of m, then it's going to grow until m plus k. Okay, and so if we take a look at sort of the graph again, right? If our cell size distribution, it looks like this. And if remember the cell size is on the bottom here, then if we just add k to the diameter of all of these, right, then we would expect this to just move over by k, right? But this is pre-division. And so the next thing that's going to happen is all these are going to divide into 2. So then we're going to get 2 cells that are n plus k over 2. And here we would get 2 cells that are m plus k over 2. And so when we divide this, then instead, it's going to narrow the scope, okay? And so you get a smaller distribution, right? And you can think about it if we want to use some numbers as an example. So if in our original graph, so we go back to the original one here with some sort of distribution like this, if our small cell here had a diameter of 4 sort of units, and here we had 6, let's say. Okay, and if we wanted to add, if our constant value was going to be 1, right, then our smallest cell will grow from 4 to 5, but divide into 2.5 diameter cells, okay? And then our larger one would go from 6 to 7, but divide into 2 3.5 diameter cells. Okay. So in this instance, with a K of 1, we've actually reduced the size here, but the spread here, the distance between these 2 points is 1, right? Whereas initially the spread was 2. Okay, so our maximum and minimum has decreased, right? And so that shows that the cell distribution would narrow. Okay, and so we can move on to this part B of question 78, and that is to design a plan to model the timer model and the more recently the constant growth, or like growing by a constant size. Okay. So with the timer model, an experiment, and certainly there's no one right answer here, but one idea that I would have is to, we're going to culture some bacteria. Okay, so we have our little plate, and we're going to get our isolated colonies, and we might take a couple of them. All right, and we'll culture them. So you could culture them in a liquid culture. So we get a couple replicates, right? And you can measure their growth at time intervals. Okay, so you have a T1, and then you come back, you have a T2. That does not look like a 2. Let's try again, T2. And you come back and you have a time 3, right? And the whole thing is, since these grow by binary fission, which means they're always doubling, right? And of course you'll have like a T0 as well, your initial. You can use these to sort of try to discern whether they are doubling at a constant pace. Okay, so if there was a timer, then after X amount of time, there would be a doubling, right? So then you'd have 2 times T0. And at some other time, then you would experience, it would double again. So you get the 4 times T0. And at some other time interval, you'd get 8 times T0, right? Because we would double again. And so you can try to discern whether you have this linear relationship from that data. Okay, and then to discern constant size. Well, one thing that's really nice about technology is we can use a microscope, right? And when you look through the lens of your microscope, okay, let that be our microscope. What we see is a field of vision that can be measured, and you can know how many units, how much big this diameter is. Okay, so we've used M and N, so let's just say it has diameter L. And from that, when you look at your image of some cultured cells, right, you can use the diameter in this image to get an approximate diameter of the cells you're looking at. And so if you take samples over time, right, we're expecting the range to narrow, right? We sort of talked about that on this previous page here. We expect the range is going to narrow, right? It went from 2 to 1. And so if we're expecting the range to narrow, then we expect their diameters to sort of become more uniform over time. So we would get our first spread of data, and then we can come back at another time and get another set of data. And you can then measure sort of their variance and see if it's increasing or decreasing. And you'd expect their variance to decrease. All right, so there's a couple ideas that I